Okay, so before I officially continue my lecture on protein folding, let's just go back briefly to the one that I discussed yesterday. May konti lang ako i-retract. Kayo na lang maghanap pagtapos ng lecture kung saan banda ha, pero may kinalaman siya sa secondary structure. Okay, may kinalaman siya sa secondary structure, just check them out. May kinalaman ito sa irregularities, irregularities ng secondary structure. Konti lang naman na modification. Di ba may na-mention tayo doon, if I'm not mistaken, merong example ng irregularity na 3:10. Napagbaliktad ko kasi, are you listening? Napagbaliktad ko kasi yung ibig sabihin ng variables 3 and 10. Tandaan, pag sinabing 3, again, pag sinabing 3, ito yung number of residues per turn. Again, it is the number of residues per turn. Whereas yung subscript na 10, again, yung subscript na 10, this refers to the intervals, ay yung pagitan, intervals ng residue, residue kung saan mangyayari ang hydrogen bonding, kung saan mag-occur ang hydrogen bonding. I repeat, for the secondary structure irregularities, there is what you call 3 sub 10. Ang 3, dapat 3 residues per turn. And then pag sub 10, it must be number of residue intervals bago mangyari or mag-occur ang hydrogen bond. Parang kahapon, baliktad yung nasabi ko. Tama ba ako sa mga nakahanap? Baliktad yung nasabi ko? Hi? Or nakita nyo ba to? Baliktad yung nasabi ko, tama? O, paki-retract na lang ha. Paki-retract na lang. So sa mga hindi pa rin makita kung saan banda ito, sulat nyo muna. Marirealize nyo siya pag inaral nyo na ha. Naintindihan? Ruwa, meron nakita mo siya? Okay, so tama yung retraction ko na pagbaliktad ko kahapon, di ba? Okay, so tama. So paki-modify na lang. Anyway, so at this juncture, thank you. At this juncture, we are going to start discussing the process called protein folding. This is defined as the process by which a protein achieves its native three-dimensional structure. I told you yesterday, again, I told you yesterday that protein folding is the process wherein a nascent, again, a nascent polypeptide is being folded or being improved para maging native conformation siya. In other means, pag sinabi nating nascent to native, we are making a non-functional polypeptide into a functional one para magkaroon na siya ng biological role inside our body. And please note that in protein folding process, this is a highly regulated, again, this is a highly regulated process, a highly regulated process. And in addition, again, this is a non-random one, okay, non-random process. So highly regulated and also non-random. So bakit natin sinabi na highly regulated and also non-random? Please note that protein folding is essential para magkaroon tayo ng normal functioning inside our body, because otherwise, kapag nagkaroon ng alteration sa folding, that could result to diseases or protein diseases, which will be tackled later. In protein folding, the process of maintaining, again, the process of maintaining or the continuous maintenance of protein synthesis and also folding is called proteostasis. So under proteostasis, there is coordination of different processes, and these processes could be categorized into 3. Again, these processes could be categorized into 3. So number 1, again, number 1, can you follow by the way? Huh? Okay, so thank you. So for the first process, that involves synthesis of nascent polypeptide, okay, synthesis of nascent polypeptide. And in other words, pag sinabi nating synthesis of nascent polypeptide, protein synthesis. And the part of the cell that produces proteins are the ribosomes, as we remember, okay, are the ribosomes. That's right. And then after that, after synthesis, of course, folding has to occur. Kaya papasok dito ang protein folding process to make the polypeptide produced by ribosomes functional para maging kapaki-pakinabang ang ginagawa ng ribosomes. Now, to be able to regulate well folding process in proteins, we have the specialized proteins that assist in protein folding, and these are called the chaperones. Kaya meron tayong segment ng discussion later na pag-uusapan natin ang chaperones. And then for the last, this involves degradation. Okay, degradation. How important is degradation? Degradation is important in case there will be misfolded proteins. Remember class, kapag meron tayong misfolded proteins, this could be a threat. MF means misfolded. Okay? Kapag kung sakali na may nabuo tayong misfolded proteins, this could be a threat para magkaroon ng pathological condition ang isang tao. Or in other words, magkakasakit siya. So to prevent that, these misfolded proteins have to be sequestered or degraded. Kaya part siya ng proteostasis. Okay? So altogether, these are the different processes involved in proteostasis. So ito rin yun. Yung naka-second bullet, ito yung summary niya. Naintindihan yung 3 steps, everybody? Okay, that's right. So with this figure, this figure shows the summary of proteostasis wherein we discuss that how many processes do we have? Ilang processes meron tayo? 1, 2, or 3? 3, very good. So first, we have synthesis of nascent polypeptide brought about by ribosomes. And the template na pagkokopyahan nun is the primary transcript or the mRNA. Hey, galing siya, of course, sa mRNA. And then, as we said, folding occurs. Okay, folding occurs so that nascent polypeptide would be converted into native proteins. Now, during the process, of course, there could be problems na pwede ma-encounter. Kaya to the rescue ang mga chaperones. Kaya every step of the way, makikita nyo na may mga chaperones na nag-act. And then finally, kung sakali na hindi naagapan ang misfolded proteins by chaperone, ang gagawin usually ng katawan is it will do its best, okay? It will do its best para ma-degrade siya, okay? Para ma-degrade. So magkakaroon ng degradation, for instance, resulting to inactive peptide fragments or okay na siya. Kaya lang, in certain situations, the body cannot cope up already with the triggers or problems. Kaya may mga pagkakataon na hindi sumasapat ang proteostasis kaya nagkakaroon ng mga aggregates. And kapag nagkaroon ng aggregates, pag hindi ito na-manage, magkakaroon ng sakit. Kaya magkakaroon ng sakit. So para lang siyang homeostasis, dito ang tawag lang proteostasis. Everybody, can you follow? Okay, that's right. O kumusta ang reception sa Zoom? Mas maganda ba kaysa Google Meet? Alam na. So eto na, okay? Pinag-aralan ko kagabi eh. Meron palang wait lang ayan. Meron palang promo ang Zoom kapag ang account mo ay educational account. So .edu.ph, parang ganyan. So tinry ko, ayan, unlimited time tayo. Okay? So alam na. Ito na lagi gagamitin. So continue natin. Pwede na tayo mag-continue, guys. Okay, so in protein folding, we discussed that protein folding happens in milliseconds. So when we say milliseconds, sobrang bilis. Kaya we can expect that in a short period of time, there will be numerous proteins that will be produced. And we discussed also that the ribosomes are the ones that produce nascent polypeptides. However, such formation is not yet enough kasi pag sinabing nascent, may kwenta o wala pa sa katawan, wala pa siyang kwenta. Okay? Wala pa siyang pakinabang sa ating katawan. That's right. So increased concentration of proteins affect the kinetics of folding. So ibig sabihin, the many or the more proteins are being produced, the greater the rate of folding. Directly proportional siya. And please note that ang synthesis process held by ribosomes of nascent polypeptide would serve as determinant if ever ang protein ba ay magiging maganda or magiging beneficial sa atin or ultimately magiging toxic or magkakaroon ng adverse effects. So kaya nakalagay dito, information needed to correct protein folding is contained in the primary structure. Ang point is, pag may mali sa primary sequencing ng polypeptide, susunod na dyan ang mga higher levels that could result ultimately to protein folding. Okay, protein folding. Now, ano ba yung pagkakatandaan nyo? Kung natatandaan kahapon anong pagkakatandaan nyo sa primary structure? Pag primary structure, ano ang characteristic bond na meron? Peptide bond lang, hindi ba? So peptide bond. Ang peptide bond ba ay rotational or hindi? Hindi. Very good. Hindi siya rotational. Meron siyang partial double bond character. Dito ba natin ika-categorize ang amino acid sequence? Yes or no? Yung sequencing ng amino acids, yes. Okay, tandaan dito naka-categorize ang amino acid sequence. That's correct. So moving forward, we have different factors that could facilitate both in folding and refolding of proteins. So pag sinabing refolding, itatama kung ano ang maling folding to prevent diseases. So first, we have native conformation of a protein is said to be dynamical or thermodynamically favored. Pag sinabi nating thermodynamically favored, this tells us that the protein folding is said to be or is a process that happens in an orderly and also guided fashion. Again, this occurs in an orderly or guided fashion. Kaya nga nabanggit ko kanina, anong sabi ko sa proteostasis? Ano yung N na sinabi ko? Non? Anong sinabi ko? Non-random. Okay, non-random siya. So it occurs in an orderly and also guided fashion. So that is synthesis, and then after synthesis, folding, and then after folding, degradation in case may problem. Next, folding is said to be modular. So pag sinabi naman nating modular, the events that happen, again, the events that happen is said to be step by step. Okay, step by step. So that is step by step. Folding is modular. So in stage 1, there is synthesis gap. A synthesis, newly synthesized polypeptide folds, and then mapapataas ang level of structure niya. And then after that, dito na papasok ang proper regulation. Okay, proper regulation. In proper regulation, to maintain normality of the produced protein, it has to be realized that the exterior part of the protein being produced is where the hydrophobic or hydrophilic residues ang nakapwesto. Pag sa exterior, para normal yung protein, ano dapat ang nandito? Polar or non-polar na mga residues? Para masabi mong normal yung nagawang polypeptide or protein. Polar, very good. Pag sinabing polar, hydrophilic or phobic? Philic, very good. So hydrophilic siya. That is in the exterior, in the exterior. And then in the interior naman, again, in the interior, ano dapat ang nasa loob ng produced or folded polypeptide or protein? That is non-polar. That is hydrophobic. Okay, hydrophobic dapat siya. I repeat, exterior, polar or hydrophilic, and then interior, non-polar and phobic. That's why you have to familiarize yourselves once more on the amino acids na may polar and non-polar side chains na tinakal natin ng lesson 1. Until such time that native conformation will then be achieved. Ito ba yung ultimate goal na folding? Yes. Kasi para ma-render nga ang protein na functional. Okay, na functional. Can we proceed on this slide, guys? Okay, so thank you. Moving on, let's have auxiliary proteins that assist folding. Remember, folding process has to be regulated because not all the time the product or the protein that will eventually be produced would be good. Kaya kailangan may nag-a-assist. Majority of the assistants are the chaperones as we discussed earlier. However, there are miscellaneous depending on their target protein such as protein disulfide isomerases and proline cis trans isomerase. So first, we have chaperones. So discuss natin what are chaperones. Di ba ginagamit din natin ang term na chaperones pag may kasakasama ka. O parang ganon ang chaperone na protein. Ang ibig sabihin ng chaperones, assistant sila ng mga proteins habang finofold. Kaya chaperones ang tawag. These are specialized group of proteins that are required for proper folding, for proper folding of many proteins or many species of proteins. So these are also known as polypeptide chain binding proteins. So they are so-called as polypeptide chain binding proteins, and they can also act as catalysts. So when we say catalysts, they can actually hasten, the chemical reactions involved in protein folding. Anong ibig sabihin ng they can hasten? Pinapabilis o pinapabagal nila? Pag-hasten? Pinapabilis. That's good. So they can hasten, pinabibilis nila ang rate of chemical reaction. So that's the role of chaperones. Now, ang chaperones ay martir. Bakit siya martir? Kasi ang chaperones, sumasama lang siya sa journey pero hindi sa final destination. Anong ibig sabihin yan? Pag kailangan siya, nakasunod siya. O sasamahan kita protein, kailangan tama ang folding mo. Pero pag na-achieve na ng protein ang correct native conformation, aalis na siya. Kasi feeling ng chaperones, hindi na siya kailangan. Okay? Kaya nakalagay dyan, do not form part of the final structure ng protein. So walang hugot doon para lang magets nyo. Okay? So it only assists during the journey, yes. Pero pag narating ang final destination, feeling niya, hindi na siya kailangan. O yung mga chaperones dyan, mag-isip-isip na kayo ha. Okay, next. After natin sa info na do not form the part of the final structure, they also suppress non-productive interactions by binding transiently. Kaya nga ang term natin ay during the process or journey lang kasi transient lang ang binding. And they were first identified as heat shock proteins. By the way, nasusundan pa ba ako? Okay. So, they were first identified as heat shock proteins. Why? Based on experiment before, tumataas ang concentration ng chaperones, kapag ang temperature ay elevated sa cell, during elevated temperature. Again, ang heat shock proteins or chaperones are said to be abundant in cells that are being stressed by high temperature environment. Kaya heat shock. Question, ang heat ba ay isang trigger for protein destruction? Yes. Okay? Kaya hanggat may iwasan ang destruction ng protein, chaperones are acting. Kaya tumataas sila at high temperature environment. That's why they were first identified as heat shock proteins. And then, heat shock protein expression is elevated. Na-discuss ko na yan. Elevated when cells are grown in higher than normal temperature. And they act through ATP-dependent mechanism. So, na-discuss natin yung figure na to kanina. You can see here, apparently, that there are chaperones that act in almost every step of the way. Because ang ultimate goal ng chaperone, ulitin natin, the bottom line is to prevent aggregation or misfolding of proteins. So meron tayong iba't ibang klase ng chaperones. Again, meron tayong iba't ibang klase ng chaperones. First stop, we have the heat shock protein 70. Ang heat shock protein 70 is often found in the cytoplasm, in the endoplasmic reticulum, in the chloroplast, and also in the mitochondria. So this type of HSP or chaperone binds to regions of unfolded polypeptides such that the hydrophobic residues will be placed interior. Ito ba ang magandang outcome kapag hydrophobic ay nasa loob? Magandang outcome ba siya pag ang hydrophobic ay nasa loob ng protein? Yes. So yan ang function ng HSP70. Usually ang inaatake nila yung mga polypeptides na maraming hydrophobic residues. Kasi pag maraming hydrophobic, mataas ang chance na sumulpot sa labas. masama yun. Kailangan itago as much as possible. So it blocks folding of proteins that should be remain unfolded and facilitates quaternary assembly of oligomeric proteins. Another type, we have chaperonins. Chaperonins are so-called as cage-like. Okay, these are so-called as cage-like chaperones. I repeat, chaperonins are so-called as cage-like cage-like chaperones. That's why, as you can see on the picture, they look like cages. Okay, they look like cages. So magkaiba ang specific term sa chaperonin depending kung prokaryote ang cell or eukaryote. Kapag prokaryote, it's GROW-EL. Pag naman eukaryote, it's HSP60. Okay, HSP60. So just take note the description of these types of chaperonin. So I repeat, cage-like chaperones, we have two types. For prokaryotes, we have GROW-EL. GROW-EL talaga yung pangalan niya. And then for eukaryotes, we have HSP60, okay? Heat shock protein 60. So take it all together, that's the end of the first auxiliary protein. Ang first auxiliary protein natin ay chaperones that assist in folding. Now, meron ba tayong nabanggit na mga miscellaneous kanina? Again, meron ba tayong nabanggit na mga miscellaneous kanina? Yes or no? Check your notes. May miscellaneous ba tayong auxiliary proteins? Di ba majority ay chaperones. So meron tayo mga miscellaneous. Ano yung mga miscellaneous? Meron ba? Choppy ba ako? Or di lang talaga kayo nagre-reply? Or nawala ba ako? Hello? Nawala siguro ako. Meron. O ba't di kayo nasagot? Ba't di kayo nasagot ha? Pag kasi hindi nasagot, feeling ko, nag-iisip. Oo nga naman. Huwag mo kaming istorbohin, sir, nag-iisip kami. Kung paano kami mananatiling gising sa oras na to. Nag-iisip pa kami. Balikan na lang natin yung slide ha. Ito yung auxiliary proteins. Naalala nyo to kanina. Di ba tatlo? Majority ang chaperones. Ito yung miscellaneous. Okay, ito yung miscellaneous. Okay na? Naalala na? Okay. So, punta na tayo sa miscellaneous. Second, we have protein disulfide isomerases. So protein disulfide isomerases stabilizes, again, it stabilizes the tertiary and quaternary structures, and usually it eliminates folding of intermediate it eliminates folding of intermediates with inappropriate disulfide crosslinks. So with that being said, we can realize that nakafocus ang protein disulfide isomerase sa mga residues na may disulfide linkages. Ano ba yung may mga disulfide linkages? Yung cysteine residues. Ay yung mga polypeptides na may cysteine residues. Because it is the one responsible for disulfide bond formation. Okay? Next, we also have proline cis-trans isomerases. So these enzymes are also known as cyclophilins. Again, these are also known as cyclophilins. And these are usually found, or they actually target the polypeptide na may X-pro peptide bond. Again, tinatarget nila ang may X and pro peptide bond. Anong meaning ng X-pro? Pro is proline. Okay, so this refers to the amino acid proline. And then X, ang ibig sabihin ng X is any amino acid. Okay, any amino acid. So anong point niyan? Again, what's the point of this X-pro peptide bond? In short, ang tinatarget ng proline cis-trans isomerase ay yung mga polypeptide na may peptide bond na katabi ng proline. Lahat ng katabi ng proline. Okay? Kahit anong amino acid pa yan, basta katabi ng proline. Doon nag-act ang proline cis-trans isomerase. So this enzyme catalyzes isomerization of the bond from being trans to cis. Okay? From being trans to cis. Kaya ang term natin ay cis-trans isomerase. Okay? Next. So overall, we are done discussing auxiliary proteins. So going back for the auxiliary proteins that assist in folding, we have three. The major ones are, ano yung majority? Anong tawag sa kanila? Chaperones, right? And then for the assistant, or let's say miscellaneous, we have disulfide and also cis trans. Okay, so take note of that. Pag proline cis trans isomerase, X-pro. Kapag disulfide, that is cysteine residues. So yun yung palatandaan natin. And then for the a last factor affecting protein folding, this is a dynamic process. So when we say dynamic process, multiple processes occur so as to maintain proper folding of proteins. That's why we discussed that chaperones do their best to be able to rescue the unfolded proteins or potentially yung mga magmi-misfold. They are always to the rescue. Okay, yun yung function ng chaperones. And then for glutathione naman, accordingly, this is the one that reduces inappropriate disulfide bonds, that could be seen, in the polypeptide, particularly those that are placed exterior. Okay, those that are placed exterior. So these are the factors. Okay, these are the factors that could affect protein folding process. Can you follow? What about the others? How are you? Are you still awake? I cannot see you, don't have video eh. Pero kita ko kayo ngayon. I mean, kita ko yung video grid. Kaya lang, wala kayong mga mukha. So, hindi ko alam kung kumusta na kayo. Ha? Pwede na tayo mag-continue? Okay. Nice. Remember, so far we were able to discuss proteostasis, particularly the process of protein folding. So kung meron tayong term na protein folding na diniscuss natin ngayon lang, ang susunod na slide ay may kinalaman naman sa unfolding. So pag sinabi nating unfolding, again, pag sinabi naman nating unfolding, ito ay kabaliktaran. That is, yung protein na functional ay nagiging non-functional. Okay? So tandaan niya na. So kung kanina diniscuss natin ang pag-convert ng protein para maging functional, ngayon yung susunod na slide na i-discuss ko, may kinalaman naman to sa kabaliktarang pangyayari wherein ang functional na protein gagawin nating non-functional. Nagets yung key concept? Nagets yung sense ng next part ng discussion? Okay, so that's why ang next topic ay may kinalaman sa term called protein denaturation. In protein denaturation, we tend to disrupt the structure of the protein such that the biological action of the protein will be removed. Mawawala siya, will be omitted. Kaya sinabi ko na may kinalaman ito sa pag-convert ng isang functional protein to a non-functional. So therefore, let me ask you, with this being said, during protein denaturation, pinapataas ba natin ang level ng protein structure o pinapababa natin ang level ng protein structure? Taas o baba? Very good. Pinapababa natin ang level ng protein structure. So kung dati kapag proteostasis from primary, secondary hanggang quaternary, ito from being quaternary, pabababain natin to tertiary to secondary until such time na magiging primary siya na ang tendency wala nang function sa katawan. Ganon ang protein denaturation. Nagets ba ang principle ng denaturation? Okay, so in protein denaturation, we tend to reduce folding of protein such that the quaternary structure, let's say, ay napupunta into its primary level, rendering the protein non-functional. However, again, tandaan, once primary structure has been reached, this is now the lowest level. Ito na lang ang pwedeng pinakamababang kahinatnan pag ang protein ay na-denature. What do I mean by that? Again, anong ibig sabihin natin sa lowest level? Pag sinabi nating lowest level, pag na-denature ang mataas na klase ng protein, ang pinakapangit, kumbaga, na structure na lang niya is sequence of amino acid residues. So sequence lang polypeptide. But take note, we cannot anymore separate all the residues through denaturation. Okay? So hindi kaya ng denaturation ang separation ng mga residues. Instead, ang magbe-break ng peptide bond ay hydrolysis. Okay? So ang magbe-break ng peptide bond ay hydrolysis. So I repeat, hydrolysis is the process of breaking peptide bonds. Siya lang ang makakapaghiwalay sa mga residues. Denaturation is limited hanggang sa sequence lang. Did you get my point? Okay, very good. That's why you can see, again, you can see here na yung isang protein sa figure sa baba, yung isang protein, complex protein na nag-curl na somehow, so may proper folding na, during denaturation, nawala yung proper folding niya. So as simple as that. Okay? So as simple as that. Next. So ito applicable sa lab, Ang susunod na question is, makinig, ang susunod na question, Sir, anong mga possible factors na pwedeng magpa-denature sa protein? Of course, usually external factor 'yon. Halimbawa, folded na yung protein, functional na yung protein, may external stimulus. May adverse stimulus from the outside kung saan na-expose ang protein. Doon siya madedenature. Ang pinaka-common, exposure to temperature. For example, pag sobrang taas ng temperature o kaya changes in pH. Tapos na kayo sa lab sa ganitong mga topics sa lab? Or not yet? Tapos na. Pero mas nakilala nyo ba kung anong nangyayari sa denaturation? Nag-experiment kayo. Pero mas naintindihan nyo yung principle niya, nagiging non-functional yung protein. Okay? Nawawalan siya ng kwenta. Parang ganon. So, ano yung mga possible factors, possible triggers para masira ang protein from its being functional? So, meron tayong iba't ibang causes. Check the next slide. May table, di ba? Yung sagot sa blank na to, yung nasa table sa next slide. So just take note the agents that can cause protein denaturation such as heat, UV radiation, acids and bases, urea, and so on. So it's your responsibility to read this table and also observe the effect. So halimbawa, magbigay ako representative halimbawa, heat. Ang heat, sinisira niya ang hydrogen bonds. Question, ang hydrogen bond ba ay importanteng force of attraction para mag-fold? Para mag-fold o mag-bend yung mga sequence? Yes or no? Ulitin natin for the benefit of everyone. Ang heat daw, sinisira niya ang hydrogen bond. Importante ba ang hydrogen bond para mag-fold ang protein o ang peptide? Yes. Di ba ang secondary structure na discuss natin may hydrogen bond? So pinapababa talaga natin. Okay? So very good. Now, ang question dito is, balik tayo, ang question dito is, ang protein denaturation ba ay reversible or irreversible? Take note, most of the time, it's irreversible. Okay? Irreversible. So, irreversible meaning, once the protein has been denatured, yun na siya. Okay? Babay na siya. Unless, meron tayong special process or special treatment na gagawin through renaturation wherein maibabalik natin siya into native conformation, but it's quite difficult. Okay? Pero generally you can classify protein denaturation as irreversible. Okay? Do you understand so far? Okay. And then, guys, pakibasa silently yung effect na nakalagay dyan. Yung effect ba, naintindihan na ba yung ibig sabihin nito based on my discussion? Based on my discussion, unfolding and disorganization not accompanied by hydrolysis of peptide bond. Okay? So, ang hydrolysis lang ang makakasira ng peptide bonds. So, next. Ito na yon, nasabi ko, commonly, irreversible siya. Kaya nga nakalagay sa reversible rare cases. Okay? Yun lang ang tandaan nyo sa part na yan. Overall, naintindihan ang protein denaturation? How about the others? Are you well? Okay pa kayo? Ayan. O, recap tayo. Sa second part on lesson 2, we were able to discuss protein folding process and protein denaturation. Now, to trigger your minds sa next discussion natin sa lesson 2, hindi ba ang proteostasis ay mahalaga para ang protein na magagawa hindi magkukos ng disease? Pero syempre, may mga pagkakataon that the body cannot anymore regulate proper folding. That's why diseases happen. In connection to that, let us tackle the different pathologic consequences of protein conformation perturbation or meaning misfolding of proteins. So meron tayong iba't ibang klase ng sakit that can be attributed by misfolding of proteins. First stop, we have the prion diseases. Actually, pag sinabi nating prion, meron siyang ibig sabihin. Ang ibig sabihin natin ng prion ay proteinaceous. Again, this means proteinaceous infections only. Again, when we say prion, it means infections only. Kaya prions. Okay? Proteinaceous, prion. Okay? Nagets niyo ba't prion ang tawag? Para siyang abbreviation. So regarding prion diseases, these are transmissible spongiform encephalopathies wherein the primary target is the brain. Kaya nga encephalopathy ang tawag. So this is actually fatal. Now, may mga variations tayo sa tawag sa mga prion diseases depending on the organism being affected. Okay? Depending on the organism being affected. So halimbawa, kasali sa prion disease ang Creutzfeldt-Jakob disease, which affects the humans, scabies affecting the sheep. Okay? And then we have mad cow disease in cattle. Ang Alzheimer's disease ay separate form siya pero prion din siya. Okay? Pero prion disease din siya. Theoretically or hypothetically, let's say. Now, regarding Creutzfeldt scabies, nasusundan pa ba ako? Nasusundan? Creutzfeldt scabies and BSE or bovine spongiform, same manifestation but it only differs which organism is affected. So halimbawa, ikaw, sinabihan ka, nako, si Pedro may scabies. Wag kang papayag kasi hindi ka tupa, tao ka, okay? Tao ka. So parang gano'n. Hindi ka pwedeng sabihin, may mad cow siya, hindi ka baka, okay? Hindi ka baka. Dapat ang tawag sa condition mo, ay social dapat. Creutzfeldt-Jakob, okay? Creutzfeldt-Jakob ang tawag dyan. Naintindihan? Okay. So regarding prion disease, ano ang reason bakit may prion disease? So may kinalaman ito, ang bottom line lang nito, may kinalaman sa misfolded proteins. Ngayon, anong klaseng misfolded protein ang involved? Again, anong klaseng misfolded proteins ang involved? Para magets ito, i-divide natin yung discussion into two. We have the first and the second. In the left, the first one is the normal. Inside our body, we have the normal protein called PRPC. Again, we have the normal protein present called the PRPC. So when we say PRPC, it means cellular prion related protein. Again, pag sinabi natin PRPC, this is cellular prion-related protein. Huwag kayong magpapadala sa pangalan kahit may prion na term, normal protein siya sa katawan. Pero related kasi siya, okay? So prion-related protein. Let's talk about the structure of the prion-related protein. It says on the slide, the normal protein PRPC has a secondary structure containing alpha helices. Okay, alpha helices, and then easily soluble siya, easily soluble, and can actually be degraded or hydrolyzed by proteases. Okay, by proteases. So yan ang characteristics ng PRPC. Now, if proteostasis is not well maintained, chances are this normal PRPC could become PRPSC. Okay, PRPSC. Ang PRPSC ito naman yung abnormal, ay abnormal prion-related protein. Abnormal siya, kaya nakalagay abnormal because it is the one that can cause encephalopathy. As we check the structure, makinig, as we check the structure, instead of having alpha helices, ang meron beta sheets. Okay, ang meron ay beta sheets. Magkaiba ba yung alpha at beta conformation sa secondary structure? Yes. Okay? Kaya yung structure ang magde-determine kung siya ba ay good or bad. Now, unlike the normal protein, this is insoluble, yung PRPSC, insoluble. So pag insoluble, kapag indigestible siya by certain solvents, anong ine-expect nyo? Sino ang nasa exterior? Sino yung nasa exterior? Is it the polar or the hydrophobic? Kapag hirap tunawin, usually anong maiisip natin? Ano yung nasa exterior? Hydrophobic or hydrophilic? Hydrophobic. Question, pag ang nasa exterior ay hydrophobic, is it good or bad? Bad. Okay, kaya problematic siya. Are you learning so far? Okay, so that is for PRP-SC. O yun lang tandaan nyo yung pinagkaiba nila. Okay? So yan yung prion diseases. So next, since nabanggit natin na ang Alzheimer's ay isang halimbawa, hypothetically, ng isang prion disease, ipaliwanag natin anong problem sa Alzheimer's. Familiar kayo sa Alzheimer's, right? Ang Alzheimer's ay isang neurodegenerative disorder caused also by misfolding of proteins. At ang tawag natin, everybody, sa misfolded protein ay beta amyloid. Okay, beta amyloid. Now, ano ba yung beta amyloid? Ang beta amyloid ay isang misfolded protein that accumulates, tumataas siya in people with Alzheimer's disease. Okay? In people with Alzheimer's disease. And also it was found out that in Alzheimer's disease, there is also low level of the neurotransmitter acetylcholine. Again, the problems in Alzheimer's based on studies, mataas ang misfolded beta amyloid at mababa ang acetylcholine. Nakikinig pa ba? Okay. So, with these two, so far, may mga gamot tayo available in the market that increase acetylcholine levels. What I'm trying to say is, may gamot pang Alzheimer's by targeting acetylcholine. Okay? By increasing acetylcholine. So check tayo dito kasi may drugs available. Pero, makinig, ang problema is sa problemang beta amyloid na mataas, wala pa tayong gamot available in the market. Okay? Wala pa tayong available in the market. So in connection to that, i-discuss natin ang simple step kung paano ba nabubuo ang beta amyloid. So ang basic step huwag nyo nang pansinin to ha, huwag nyo nang pansinin. Doon tayo mag-focus sa beta amyloid, okay? So doon tayo mag-focus sa beta amyloid. Ang beta amyloid ay nanggagaling sa tinatawag na amyloid precursor protein. Usually, ang symbol ng protein, para lang aware kayo, CHON. Kasi ang mga proteins, may carbon, may hydrogen, may oxygen, may nitrogen. Okay? So, amyloid precursor protein. Now, ang amyloid precursor protein, hindi ko sure, baka di na ako nasusundan na, ulitin natin. Amyloid precursor protein ay nagiging beta amyloid through an enzyme. Okay? So, may enzyme na kailangan. Ang tawag sa enzyme na yon ay beta secretase. So, ang tawag sa enzyme na yon, hindi masyado malinaw, pero ito yung spelling, beta secretase. Yan, beta secretase. So, this enzyme is responsible for the conversion of amyloid precursor protein into beta amyloid. So, if that is the case, please listen. If that is the case, please answer my question. If you are a scientist and you want to discover a drug that would lower beta amyloid, do you think blocking the enzyme beta secretase is a good goal or is an ideal goal? Yes or no? Yes, okay, very good. In Tagalog, kung ikaw ay magdi-discover ng gamot pang Alzheimer's na magpapababa ng misfolded beta amyloid, pwede kang mag-discover na nagbablock ng beta secretase. Kaya marami tayong thesis or thesis, marami tayong mga researches na kinoconduct na ang goal ay targetin ang beta secretase, pwedeng gamitin sa Alzheimer's. Kaya pag nagbasa kayo ng mga studies at nakita nyo na beta secretase inhibition yung action ng compound, pwede mong isipin na ang gustong iparating ng study pang anong sakit? Alzheimer's. Kasi wala pang nagta-target ng beta amyloid na established worldwide. Okay? Gets ba? Gets? Okay, very good. Next example, hindi na to prion disease ha, hindi na to prion, pero epekto ng misfolded protein. Okay, epekto ng misfolded protein. Next, we have beta thalassemia. Beta thalassemia is a collective term pertaining to genetic defects that impair the production of one subunit of hemoglobin. Sa susunod na lesson, which will be discussed also today, ang hemoglobin ay nagke-carry o nagta-transport ng oxygen. So mamaya maiintindihan. Di ba ang no elementary tayo at high school, ang alam lang natin, hemoglobin transports oxygen. Pero sa biochem nyo maiintindihan paano yun nangyayari. Okay? So explain natin yan mamaya. And ang problem sa beta thalassemia, since defective ang hemoglobin, may problems sa oxygen transport. So that is because of the presence of ASHP, causing cytotoxicity of developing red blood cells. So that is beta thalassemia. We also have sickle cell anemia. Again, we also have sickle cell anemia. Ganito kasimple ang sickle cell anemia. Ang sickle cell anemia, sa polypeptide sequence, yung glutamic acid na residue napalitan ng valine. Okay, napalitan ng valine. So I repeat, ang problem sa sickle cell anemia, yung protein na sequence, yung polypeptide sequence sa hemoglobin, instead of glutamic acid, ang pumalit valine. O di ba, mapapansin nyo, by the way, nakakasunod pa ba? Nakakasunod pa? Ha? Okay pa kayo dyan? Okay. So, kung mapapansin nyo, yung residue, isang pirasong residue lang ang nagbago. Yung glutamic acid naging valine. Primary level lang yon. Nagkamali ng isa, domino effect na siya hanggang highest level. So kung mapapansin sa picture, ang normal red blood cell na dapat glutamic acid ang isang residue na ganito ang itsura, biconcave dapat, naging crescent shape siya dahil lang sa pagpalit ng valine. O gano'n na nangyari. Now, anong implikasyon pag naging crescent-like shaped yung itsura ng red blood cell? Hindi na siya makakapag-carry ng oxygen. Pag hindi siya nakapag-carry ng oxygen, magiging anemic yung tao. Kaya sickle cell anemia ang tawag. Okay? Gets ba? Okay. Ano dapat ang normal na hemoglobin? Anong sequence? Glutamic or valine? Ano ang glutamic acid or valine? Glutamic acid, good. Tawag dito, ano yung napalit? Valine. Okay, so ito yon. Yeah, okay, ito yon. So next, I think this is the last slide for this lesson. So other important proteins, blood sugar level in the body is being regulated by a protein, and that is a hormone called insulin. Okay, so that is insulin. So take note of the effects of insulin. Don't worry, may specific module tayo na i-discuss natin ang insulin sa metabolism. Doon natin siya i-discuss. So sa ngayon, tandaan nyo, ang insulin can cause cellular glucose uptake resulting to hypoglycemia. Okay, resulting to hypoglycemia. So altogether, we have the proteins, the level of structure, and also the proteostasis, protein denaturation, and diseases. So yan ang nag-encompass sa lesson 2. So end of lesson 2. Nagets ba overall ang lesson 2? Okay pa kayo? Okay pa? O sige. So it's already 2:02. Okay, 2:02. So start tayo sa proteins. May copy na kayo nito? Okay, so start tayo. So pansin nyo, ang topic 1, amino acids. Topic 2, polypeptides folding. Topic 3, proteins, pinaka-complex na. Okay? So palaki na ng palaki. Guys, interview lang. Interview. Compare lipids from proteins. Ano mas exciting para sa inyo? O none? O none of the above? Ano ang mas exciting for you na biomolecules so far? Proteins. How about the others? Sabi sa inyo, madali lang lipids, di ba? Ang dali lang niya. Ang proteins, masarap siyang pag-aralan pero palalim kasi siya ng palalim. Ayun. So after nito, enzymes naman, tapos nucleic acids, mako-compare nyo ano yung mga mahihirap intindihin na mga lessons. Okay? So start na tayo sa complex or biological proteins. Okay. For lesson 3, na ang goal ko ay matapos this afternoon, we are going to discuss complex proteins or biologically active proteins. The main focus of this lesson is to discuss two most dominant, two most commonly observed proteins, and these are the fibrous proteins and the globular proteins. When we talk of fibrous proteins, these are important to provide tensile strength, strength, a tensile strength, and also flexibility. Again, these proteins are important because they provide tensile strength and also flexibility. What about for globular proteins? Talking here on globular proteins, these are essential for metabolism. Okay, these proteins are essential for metabolism. Also important din siya for transport purposes, transport. And lastly, storage. And then lastly, we have storage. So we have metabolism, transport, and lastly, storage. So yan yung mga functions ng fibrous and globular. Under fibrous, we will discuss three types or three examples rather, collagen, elastin, and keratin. Whereas for globular, we have myoglobin and also hemoglobin. So with that, we can start discussing the first fibrous protein, that is collagen. Collagen is an example of a structural protein, and in fact, this is considered as the most dominant or most abundant protein in the body. Basically, pag sinabing most abundant, pinakamaraming protein sa ating katawan. For example, we can find this in different connective tissues like in tendons, in cartilages, in the matrices, and also in the cornea. So that's why ang collagen ay fibrous protein kasi siya ay nagpo-provide ng flexibility and also strength. Okay? So that is collagen. Now, ang collagen ay isang klase ng protein. That's why it is essential for us to know the structure of collagen. Again, the structure of collagen. Number 1, ang itsura ng collagen, pag tinignan nyo siya, meron siyang triple helical structure. Again, meron siyang triple helical structure. That's why in the figure, you can see that there are three chains that are found or that are intertwined with one another. The one in color blue, the one in color pink, and the one in color green. Tatlong piraso na nag-helix, okay? Nag-ikutan sila. Now, anong tawag natin, again, anong tawag natin sa bawat piraso na chain na nakahelix dito? Ang tawag natin dyan is alpha chain. Again, ang tawag natin dyan sa individual chain ay alpha chain. Now, ano ang characteristics ng alpha chain? Number 1, ang alpha chain ay isang superhelical structure. Again, isa siyang super helical structure. So I reiterate, ang alpha chain ay isang superhelical structure. So when we say superhelical, ganito siya kasimple. Ang superhelical, ibig sabihin, yung pagliko, yung pag-coil niya mas madalas. Yung pag-coil niya ay madalas. That is, ika 3.3 residue per turn siya. Okay? Again, 3.3. Ito na pala, nakasulat na. So we have 3.3 residues per turn bago liko. So halimbawa, ito yung helix drawing natin. 1, and then liliko siya after 3.3. Kaya ang tendency, mas madalas yung pag-twirl niya, okay? Yung pag-ikot niya. Kaya super helical siya. Can you follow? So far, can you follow? Okay, what else? Baka malito kayo, mga bata, baka malito kayo, hindi ba may diniscuss tayo last time na term na alpha helix? Huwag kayong malilito, ang salitang alpha chain at alpha helix ay magkaiba. Magkaiba sila. Magkaiba kasi unang-una, yung dalas ng pag-coil o dalas ng pag-ikot ay magkaiba o nagdi-differ sila. Remember ang alpha helix, tuwing kailan nagkakaroon ng turn. Tuwing kailan nagkakaroon ng turn. 3 point, 3 point, very good, 3.6. Therefore, ano siya, less or more helical? Less, very good. Kaya super helical yung alpha chain. Nagets ba na magkaiba ang alpha chain at alpha helix? Ha? Nagets? Okay, nice. Next, when we check, so yan yung secondary structure niya. Now, when we check the primary structure, pag sinabi ko ang primary structure, dapat matik sa utak nyo kung anong i-discuss natin. Pag primary structure, anong i-discuss natin? Amino acid sequence. Okay, amino acid sequence. Pag chinek natin ang primary structure ng alpha chain, each alpha chain, meron siyang format o template na sequence. Ito lang ang sequence niya, paulit-ulit lang. So we have gly, X, and Y. When we say gly pertains to glycine. Can you follow? Again, gly pertains to glycine. Ang glycine class, again, ang glycine, ang simplest amino acid as we remember, and because of the presence of only hydrogen atom in the side chain, this gives flexibility in the alpha chain. Okay, so it gives flexibility. So in short, ang purpose niya ay flexibility. Next, what does X represent? Again, what does X? Okay pa kayo? Okay pa kayo? So what does X represent? It represents proline. O sulat nyo, mag-aalbit lang ako. Proline. So X here is a variable pertaining to proline. Again, it pertains to proline. Now, what's the importance of proline naman? Proline gives kinks. Again, proline provides kinks in the structure of the alpha chain. Again, it provides kinks. Okay? And finally, we have Y. Ang Y could be HYP or HYL. So pwede siyang HYP or HYL. So when we say HYP or HYL, that means hydroxyproline or hydroxylysine. Again, it means hydroxyproline, hydroxyproline, and then hydroxylysine. So again, these are hydroxyproline, and also we have hydroxylysine. Question, based on your knowledge on lesson 1, are these amino acids standard or derived? Standard, very good, derived. Pag sinabing derived, kasali ba sa 20 o galing sa 20? Galing. Very good. At least you are learning. Okay? So, ang hydroxyproline at hydroxylysine ay derived amino acids. Now, ano naman ang importance ng hydroxyproline at hydroxylysine? They provide tensile strength. Again, they provide tensile strength. So, yan ang importance ng hydroxyproline and also hydroxylysine. So, ang point dito is, kahit gaano kahaba ang collagen, paulit-ulit lang yan. We have glycine, proline, pinaka-common hydroxyproline ha, pinaka-common hydroxyproline. So, glycine, proline, hydroxyproline, glycine, proline, hydroxyproline, paulit-ulit ganon. Okay? Hanggang makuha yung tamang length ng collagen and then mag-helical silang tatlong alpha chains. Naiintindihan ba ako? Okay. So another figure that tells us the structure of collagen, we have the amino acid sequence, paulit-ulit, ano yung X? Ano yung X? Ha? Ano yung X? Kahit letter lang, okay na. Okay. Proline, ano yung Y? Hydroxyproline, mas common. And then XY. Ano purpose ng glycine? Flexibility. Ang X, ang purpose ay kinks. Okay? And then Y, tensile strength. And then for the secondary structure of the alpha chain, sabi natin, super helical siya. Kasi nagkakaroon ng coiling tuwing gaano kadalas? Gaano ang frequency niya? 3.6 or 3.3? Okay, 3.3. Less or more helical? More. Very good. More helical. That's why in the triple helix, nagsama-sama na silang tatlo. Now, may nakalagay dito na nutritional value. Anong ibig sabihin nutritional value? May mga kinakain tayong pagkain na collagen rich. Halimbawa, gelatin. Alam niyo yung dessert na gelatin? So yung dessert na gelatin o kaya yung capsule na may gelatin, ang laman lang nun, tatlong amino acid. We have glycine, proline, hydroxyproline. Okay? Ano pa? Marami mang supplements na may collagen na nagkiklaim na nakakaganda ng ating balat. Marami ba? Uy, may collagen yan. Inom ka, gaganda balat mo. Ganito siya, okay? Hindi siya totoo. Hindi siya totoo. Kasi, ganito siya ha. Guys, nakikinig kayo? Nakikinig? Okay. So, balikan natin ang amino acid sequence. Amino acid sequence. Ang amino acid sequence ng collagen, tatlo lang. Yan lang talaga sila. Okay, yan lang talaga sila. Plus, kung ang claim ng collagen ay it should be taken by mouth, try to imagine, pag taken by mouth, sa ating sikmura, ang daming hydrolytic enzyme. Ang daming hydrolytic enzyme. Pag sinabing hydrolytic enzyme, anong reaksyon ang kayang gawin? Hydrolysis. Okay, hydrolysis. And this hydrolysis can destroy the bonds. Kaya masisira at masisira siya. Kaya pag wag kayong bibili. I mean, wala akong ibig sabihin na, pero I'm just teaching you. Yun. Naintindihan? So, hindi full yung claim. Hindi full yung claim. Okay? O, sabi ni Rose, uminom daw siya ng collagen. Anong sabi sa bawal judgmental? Hindi kami naniniwala. So, ibig sabihin, pakita mo. So, yun. So, yun. Yan yung nutritional value. O, next. Next. Pwede na tayo mag-continue, everybody? Ha? Okay. Naaalala ba ang hydroxyproline? Naaalala ba siya? Yes. So, ano to? Derive or standard? Derive. Very good. Derive. Importante ba siyang composition ng collagen? Yes. Ano ang effect niya? F, flexibility ba, tensile strength, or whatsoever? Ano ang kanyang effect? Tensile strength. Good. So ngayon, i-discuss natin paano nagagawa ang hydroxyproline. Apparently, ang hydroxyproline, makinig, hydroxyproline, ay galing sa standard amino acid na proline. So galing din siya sa standard amino acid na proline. And this derived amino acid is being produced because of hydroxylation reaction of proline residue. Now, ang responsible enzyme for the hydroxylation of proline is what you call prolyl hydroxylase. Again, that is the enzyme prolyl hydroxylase as shown in the figure. Okay, again, prolyl hydroxylase. So with that, makinig, again with that, listen carefully. So ang point dito is from proline via the enzyme prolyl hydroxylase, that causes formation of hydroxyproline. Okay, so that causes formation of hydroxyproline. However, again, listen, however, para maging active ang prolyl hydroxylase, kailangan merong cofactors. Okay? Kailangan may cofactors. Sa madaling paraan ng pagpapaliwanag, pag sinabi natin cofactor, ito ay nagsisilbing ally, siya ang ibinibigay sa prolyl hydroxylase enzyme upang ang prolyl hydroxylase ay maging aktibo para siya ay ma-activate. So meron tayo mga known cofactors for prolyl hydroxylases activation. So una is oxygen, which is very easy to acquire. Hihinga lang tayo, mag-inhale lang tayo, may oxygen na tayo. Okay? Pangalawa, we have ferrous ion, ferrous ion. And then finally, vitamin C. Vitamin C. Ang vitamin C ay tinatawag ding ascorbic acid. Ay tinatawag din siya ascorbic acid or tinatawag din siya sa inorg, di ba? Narinig nyo na siguro yung sevitamic acid. Tama ako? Narinig nyo na siguro yan. So again, ang ascorbic acid is also sevitamic as in cetos, sevitamic acid. So for the allies or cofactors, we have oxygen, ferrous. Ferrous is 2+, right? And then vitamin C or ascorbic acid, sevitamic. So with the presence, are you still listening? So with the presence of these cofactors, prolyl hydroxylase can now catalyze proline-forming hydroxyproline. That's why there is enough strength in the collagen. Okay, there is enough strength or tensile strength. Nagets ba yung production ng hydroxyproline? Ha? Gets? What about the others? How are you? Ano na napapanaginipan nyo? Okay. Now, common vitamin ba na iniinom natin ang vitamin C? Yes, common siya na iniinom natin na source ng vitamins. Marami tayo mga drugs na available sa market containing vitamin C. Ngayon, sa sobrang dami ng mga commercially available vitamin C, kanya-kanyang pakulo kung paano siya ibebenta. May iba na to boost your immunity, which is really true, really correct. Pero may isang brand ng vitamin C na matalino ang pagkaka-market. Yung brand na to, Potency. Ang commercial model si Bea Alonso. Ano yung minomodel ni Bea Alonso during that time? Sabi niya, pag uminom ka ng Potency, nakakaganda ka. Nakakaganda siya ng balat. Very true. Tama siya. Kasi ang vitamin C na nasa Potency ay isang cofactor para dumami ang hydroxyproline. Nagpapatibay siya ng collagen. Okay? At pag tumibay ang collagen, babanat ang balat, gaganda siya. Kaya yung claim na nakakaganda ang vitamin C, totoo siya. Ito yung reaksyon involved doon. Naintindihan? Hoy. Hoy mga bata, naintindihan nyo? Okay, so very good. So, it stabilizes the triple helical structure of collagen. So, okay na yan. And then, pag table, alam na, kakabisaduhin. Okay? So, this table states the different types of collagen and then kung saan parts ng katawan natin siya makikita. That's why we are very we are confident enough to know that collagen is really the most abundant protein in the body kasi may iba't ibang types found in different tissues. Now, palalimin natin ang discussion ng collagen. At this point, let us discuss biosynthesis of collagen. So in biosynthesis, ang goal natin is maintindihan nyo paano nagiging triple helical structure ang collagen. Yun ang i-explain natin dito. We can divide the steps into 7. So starting with pro-alpha chain formation until cross-link formation. Don't worry, iisa-isahin natin siya. Kaya from time to time, check this part of the slide or of the PowerPoint para guided kayo. This figure summarizes the different steps involved in collagen formation. So starting with the gene that encodes for collagen until such time na may cross-linking. So ito ay summary din na iisa-isahin natin. Okay, iisa-isahin natin. So right now, i-discuss na natin ang mga specific steps. So una, the first step involved is the production of the so-called pre-pro-alpha chain. Again, that is production of pre-pro-alpha chain in the ribosomes. Okay, in the ribosomes. So how does that happen? How does that happen? Suppose, o makinig, suppose this is the nuclear membrane of the nucleus. Okay, halimbawa, ito ay yung nuclear membrane ng nucleus, and take note that inside the nucleus, mRNA is being produced. Okay, mRNA. Now, ang mRNA, siya yung magiging transcript, siya yung magiging basehan para gumawa ng protein. And in this case, gagawin collagen. So ang mangyayari is, this mRNA produced will go out from the nucleus via the nuclear pore. Okay, lalabas siya via the nuclear pore hanggang makarating siya sa ribosome. So ang mangyayari dyan is, translation will happen. A translation will occur. So when we say translation, yung mRNA transcript ay magiging kopyahan para makagawa ng polypeptide. At ang tawag sa polypeptide na yon ay pre-pro-alpha chain. Okay, pre-pro-alpha chain. Again, that polypeptide is called the pre-pro-alpha chain. So ito siya, yung may color red at color blue. O, idodrawing ko dito ha, yung may color red at may color blue. Talagang magkaiba yung kulay niyan. So collectively, makinig, collectively, ang tawag dyan ay pre-pro-alpha chain. Now, bakit magkaiba ang kulay? So meron tayo, pag inobserbahan natin ang pre-pro-alpha chain, meron tayong side na polypeptide, yung color red, and then yung color blue naman, ang tawag dito ay signal peptide. Again, yung color blue, ang tawag ay signal peptide. Kaya ang tawag natin, as you can see here, yung color blue, may nakasulat na signal peptide. Okay? So ang tawag overall, pre-pro-alpha chain. Question? Question so far sa first step, basta nagawa ang pre-pro-alpha chain. Sa ang part, that is in the ribosome. Next, pwede na ako mag-continue? Ha? Okay. Now, i-highlight natin ang signal peptide. Okay, signal peptide. Tandaan, as a general rule, essentially speaking, kapag ang isang polypeptide chain ay may signal peptide, ang nagiging fate o nagiging kapalaran ng polypeptide pag siya ay nagawa, ilalabas siya. So, exits the cell. So, ultimately, ang mangyayari dito is yung isang polypeptide na may signal peptide ay eventually lalabas ng cell. So ang punto dyan, makinig, ang punto dyan, pag walang signal peptide usually, naiiwan lang yung peptide sa loob ng cytoplasm ng cell. Nasa loob lang siya ng cytoplasm. Okay? Kapag walang signal peptide. Pero pag may signal peptide, para siyang driver, o ilika dito, lalabas tayo ng cell, hindi ka pang loob lang. Okay? Yun yung ibig sabihin ng presence ng signal peptide. So with that, anong susunod na mangyayari? Ang palatandaan nyo sa signal peptide, driver nga siya. Siya yung mag-guide doon sa original peptide chain para ilabas ng cell. Pero hindi siya basta-basta. Siyempre, may iba pang mangyayari. So next step, number 2, a signal peptide directs them first to the rough ER. Okay? It will direct sa rough ER para mabuo ang pro-alpha chain. So kanina, ang tawag ay pre-pro-alpha. Yung ngayon ang tawag ay pro-alpha. How does that happen? For instance, the totality is the pre-pro-alpha chain, right? Yung total structure. Ngayon, pagpasok ng polypeptide sa rough ER, makikleave, matatanggal, mapuputol ang signal peptide. When the time comes that the signal peptide is cleaved off, ang tawag sa natira, pro-alpha chain. Gets? Pag may signal peptide, anong tawag? Pre-pro o pro lang? Pag may signal peptide, pre-pro, very good. Kapag wala, anong tawag? Pro, okay? Pro-alpha. Moving on. So imagine nyo ha, nagawa ang pre-pro-alpha chain at yung driver, ginaid siya papuntang rough ER. Pagpasok ng rough ER, natanggal yung driver. So ang natira, pro-alpha chain. Kaya sa rough ER na, nasa loob na ng rough ER, yung pro-alpha chain. Yung pro-alpha chain. Now, anong nangyayari dyan? Ang nangyayari is, yung isang part, yung isang residue ay ini-improve o kino-convert into a stronger version. So that involves hydroxylation already. Hydroxylation of lysine and also proline. So that is, for lysine, for instance, via the enzyme lysyl hydroxylase, that causes formation of hydroxylysine. And then yung proline na residue naman, ginagawang hydroxyproline by the prolyl hydroxylase. Question, may cofactors needed ba dito kay prolyl hydroxylase para ma-activate? Yes or no? Kailangan ba natin ng cofactors for prolyl hydroxylase? Yes or no? Ha? Hi? Nawala ba ako ulit? Nawala ba ako? Sorry. So ulit, kailangan ba ng alay? Ito yung sinasabi ko, kailangan ba ng alay ng prolyl hydroxylase para siya ay maging active? Yes. Okay, yes. Kaya nagiging hydroxyproline siya. Nangyayari yon sa rough ER. Ano-ano yung tatlong kailangan? O, ano yung O? Oxygen. Ano yung F? Iron. Ano yung vitamin na kailangan? Anong letter? C. Okay, vitamin C. That's right. So that's why yung third residue nagiging derived amino acid. Next, step 4. O recall tayo sa mga steps. Step 1, ano nagagawa? Pre-pro-alpha chain. Step 2, pro-alpha chain. Step 3, derive amino acid formation. Okay, derive amino acid formation. And then step 4, glycosylation. So simply pag sinabi natin glycosylation, there is carbohydrate addition in the structure. A carbohydrate addition in the structure in some hydroxylysine residues. So usually, ang nalalagay is either glucose or galactose. Okay, so it can be glucose and galactose. Kaya nakalagay dyan galactosyl for galactose, glycosyl for glucose. Next, step 5. O proceed tayo sa step 5. So lahat ng diniscuss natin kanina sa rough ER nangyayari so far. Yung pag-hydroxylate at glycosylate. Now, ang susunod na mangyayari is assembly of 3 alpha chains to form pro-collagen. So this involves the formation of disulfide bonds between C-terminal extensions of pro-alpha chains. So what do we mean by that? Okay, dagdagan nyo to ha. Assembly of 3 pro-alpha. Okay, pro-alpha chains. Okay, so dagdagan nyo na salitang pro. So sa number 5 is assembly of pro-alpha chains na nangyayari sa Golgi apparatus. I repeat, na nangyayari sa Golgi apparatus. So continue natin. Anong ibig sabihin ng fifth step? So in the Golgi apparatus, ang nangyayari dito is yung tatlong pre-pro-alpha chains daw ay nag-a-assemble, nagsasama-sama sila. Okay, nagsasama-sama sila. And then, distinct dito ang presence ng disulfide residues sa C-terminal portion. Okay, sa C-terminal portion. And because of the presence of disulfide residues in the C-terminal portion, nagiging effect is nafo-form ang pro-collagen. Okay, nafo-form ang pro-collagen. So anong appearance ng pro-collagen? Ganito ang itsura ng pro-collagen. Sa gitna, sa center, siya ay helical. So we have 1, halimbawa 2, and then 3. Ay halimbawa 3 yan. So sa gitna, sa center, helical yung structure. So nag-ikot-ikot na sila sa gitna. Pero tandaan, sa gilid, diretso pa rin. Okay, sa dalawang gilid or sa lateral portion, non-helical pa rin siya. Okay, non-helical. So, ganyan yung appearance. Okay? So, pro-collagen ang tawag dyan. So, ikot-ikot sa gitna, sa dalawang gilid, diretso pa. Okay, diretso. So, that is pro-collagen. Pero at least nakita nyo na meron ng ikot-ikot na nangyayari pero hindi pa kumpleto kasi may diretso pa sa dalawang gilid. Okay? That's why it says here, procollagen is the assembly process aligns or in procollagen formation, the assembly process aligns three alpha chains relative to one another. The three alpha chains are wound around with one another. So pag sinabing wound around, nag-ikot-ikot. But, again, but yung nasa dulo, diretso. Ang tawag natin sa non-helical portion, ay registration peptides. Again, ang tawag natin dyan ay registration peptides. Okay? So, natapos natin so far ang step 5. So, recap tayo ng mga steps. Hindi pa tapos lahat-lahat ng steps, recap muna, review. So, una, continue natin from hydroxylation and glycosylation. Anong sabi natin? After hydroxylation and glycosylation in the rough ER, pupunta siya sa GA. Ano yung GA? Anong part ng cell ang GA? Golgi apparatus. Sa Golgi apparatus, ano nagagawa? Pro-collagen. Question, sa pro-collagen ba guys, lahat ba ay nakaikot na o may straight pa sa gilid? Meron pa bang straight? Yes, very good. Anong tawag sa mga straight portion sa gilid? Anong tawag doon? Registration peptides. Very good. Kaya itsura niya dito, hindi pa siya fully wounded with one another. Okay? So next, step 6 na so far, step 6. Ang step 6, it involves exocytosis. Okay, exocytosis. So when we say exocytosis, halimbawa Golgi apparatus to, ang tendency is ipapak through vesicle, ang pro-collagen. Okay, ipapak sa vesicle ang pro-collagen and then ready na siyang ilabas through exocytosis. Kaya nakalabas na siya. So at the end of step 6, we can tell that pro-collagen immature pa to. Pro-collagen is found in the extracellular matrix. So in step 7, sa extracellular matrix, ang nailabas na pro-collagen will be catalyzed by an enzyme. And that is called the pro-collagen peptidase. So first stop, para maintindihan nyo ito, pag sinabi natin na peptidase, isa itong hydrolytic enzyme. Okay? Isa itong hydrolytic enzyme. So when we say hydrolytic enzyme, this tells us that peptidases promote hydrolysis. Okay, hydrolysis. Question, pag may hydrolysis, nasisira ba ang peptide bond? Yes or no? Pag may hydrolysis, nasisira ba ang peptide bond? Yes, very good. That's why in the figure, wag niyong pansinin yung dulo dapat diretso to. Mali yung nasa drawing. Dapat diretso tong nasa dulo. Yung registration peptides. Yung nasa gitna ang kulubot, okay? Or yung nakaikot. So, ang gagawin ng pro-collagen peptidase, sisirain niya ang peptide bond na nagbe-bridge o nagli-link sa helical at non-helical portions. Ulit, ang ginagawa ng peptidase dito, yung nagko-connecta sa non-helical at helical portion. Sa non-helical at helical portion, pinuputol. Kaya pansin nyo, anong natira sa gitna? Yung helical o non-helical? Anong natira sa gitna? Helical or non-helical? Helical. So naputol. Okay, naputol na siya. Nagets nyo ba? Nagets nyo? Okay, anong tawag sa naputol? Tropocollagen. Okay? Ang tawag ay tropocollagen. Medyo confusing yung pangalan, di ba? Pre-pro-alpha, pro-collagen, tropocollagen. So please observe the names. Okay? Observe the names. Next, number 8. Number 8 is the self-assembly or polymerization of collagen molecules forming fibrils. So in this case, there is overlapping of the neighbor by a length, approximately 3/4 of a molecule. So as you can see here, so may overlapping yung mga tropocollagen forming fibrils. So nagne-networking na sila, nagkakaroon na ng interconnections. Interconnections would lead to further strengthening of the collagen. Okay, so yan yung self-assembly or polymerization. And then, for the last, we have cross-link formation. So in cross-link formation, to be able to facilitate this, thus making the collagen more stable and more stabilized, we need the enzyme lysyl oxidase. And just like prolyl hydroxylase kanina, ang lysyl hydroxylase kailangan din ng cofactor. So ang kailangan ng cofactor is copper. Okay? Copper naman yung kailangan. So ulitin natin, ang kailangan ay copper. So take note of that. Okay? Next. So ganito yung discussion natin kanina, i-summarize lang. Naaalala nyo yung almost straight na hindi nag-helical structure? Sila yung natatanggal. Both the N-terminus and the C-terminus of the peptide. Kaya tendency ang natitira, yung nasa gitna. Anong tawag sa gitna? Anong tawag dito? Tropocollagen. So ang tawag dito ay tropocollagen. And this tropocollagen would cross-link and then ultimately forming the final product that is strong, that is flexible, and that would resist breakage. That is yung final product na collagen. So diyan natatapos ang synthesis of collagen. Another figure that would summarize all of the processes, or recap lang to. Bago tayo mag-recap, na-gets nyo ba yung step by step? Ha? Na-gets nyo? O reviewin natin ha. And then after this, meron tayong short break. Bago tayo mag-continue. So for the review on collagen synthesis, di ba nasabi natin, may mRNA, ito yon, may mRNA, and then saan pupunta? Pupunta sa ribosome, and then yung ribosome, ang tendency, magpo-produce ng pre-pro-alpha chain, and then pupunta sa RAF-ER. O ano nangyayari sa RAF-ER kanina? Anong sabi natin? Sa RAF-ER, nagkakaroon ng hydroxylation. Ito yon, di ba nakita nyo? Hydroxylation. And then, nagkakaroon din ng glycosylation. Okay, nagkakaroon din ng glycosylation. And then, anong susunod na mangyayari? Pagpunta sa Golgi apparatus, magkakaroon ng formation ng pro-collagen. Yung pro-collagen, anong characteristics ng pro-collagen? Lahat ba ay helical o may parts lang na helical? May parts lang na non-helical. And ang non-helical ay nasa gitna o nasa gilid? Ulit, non-helical, gitna o gilid? Alin ang non? Non-helical. Non. Gitna or gilid? Gilid. Kaya nakalagay dito, nakaturo, tingnan nyo ha, nakaturo with non-helical properties. And then, marerelease via exocytosis. Marerelease via exocytosis. And then, pag na-release via exocytosis, ano mangyayari? May tatarget bang enzyme doon sa pro-collagen? May tatarget ba na enzyme? Yes. Anong gagawin ng enzyme? Puputulin niya. Ano ang puputulin niya? Yung nasa gilid. Okay? Yung puputulin niya yung nasa gilid. Kaya nga, peptidase yung tawag. And therefore, magkakaroon ng tropocollagen. Okay? So that can cause formation of tropocollagen. And then, pag may tropocollagen, meron ng cross-linking. Kaya, strong na ang na-form na collagen. So that concludes our discussion on the synthesis of collagen. Naintindihan? Ha? Okay. So let's have a 5-minute health break sa mga naiihi. Kasi sabi nung iba, konting ihi lang nila, marami na silang na-miss. O sige, bigyan natin ng time yung pag-ihi. So 5-minute break. So please be back at 2:45. 2:40 sa akin eh. So 2:45 ha. I'll see you later. Ayan, continue ko na recording. Pwede na tayo mag-start. Okay pa ba kayo? Promise yan ha. Alam ko nakakapagod yung biochem, pero malapit na mag-time. Excited din ako mag-time eh, huwag kayong mag-alala. Medyo lutang na, oo nga eh. Nanginayan kasi ako sa time. Baka sabihin nyo, yung oras binayaran niyo sa CEU. Sayang na may matututunan nyo, di ba? O sige, babay na. O, dismiss na tayo. Sige, totoo na to. Mag-continue na tayo. So, after nating i-discuss ang structure at ang synthesis ng collagen, dapat nating maintindihan na may mga conditions tayo that are related to collagen abnormality. Meron tayong sakit sa katawan, may mga possible conditions in the body wherein the problem is abnormal synthesis of collagen. Collectively, these are called as collagenopathies. So the following are the examples. Maiintindihan nyo ang mga sakit na to pag nagets nyo yung diniscuss ko kanina. Number one is osteogenesis imperfecta. This is also known as brittle bone syndrome wherein ang mutation involved is nare-replace ang glycine with any amino acid with bulky side chain. Question, is presence of glycine essential for the formation of collagen? Sa sequence? Yes. So it provides flexibility because of its side chain. So pag napalitan siya ng bulky side chain, magkakaroon ng abnormality sa collagen produce. Kaya very important yung diniscuss kong synthesis kanina. We also have Ehlers-Danlos syndrome. For Ehlers-Danlos syndrome, this is characterized by inheritable defects in collagen molecule in which ang defective genes are the ones that encode alpha collagen 1, pro-collagen N peptidase, and then lysyl hydroxylase. Remember, these enzymes are important like pro-collagen peptidase and then lysyl hydroxylase sa production ng collagen. Anong importance ng pro-collagen and peptidase? Di ba ang peptidase, siya yung nagtatanggal ng registration peptide sa gilid. Naalala niyo yung kanina? Important siya for the process. And then lysyl hydroxylase is also important for hydroxyproline formation or hydroxylysine formation in case. So that's why pag merong abnormality sa mga enzymes na yun, that could result to Ehlers-Danlos syndrome. Moving on, we have Menkes syndrome. Regarding Menkes syndrome, this is also known as kinky hair syndrome, a kinky hair syndrome. At ang problem naman dito ay yung lysyl oxidase enzyme, okay? Lysyl oxidase. Why? Because in Menkes syndrome, the one that decreases, the one that depletes is copper. Okay, copper. Question, important ba ang copper for lysyl oxidase activity? Yes. So absence or deficiency of copper would affect oxidase's action, thus maapektuhan ang final cross-linking process. So in Menkes syndrome, this usually affects boys or males, and it affects copper levels and metabolism in the body, causing seizures, brain damage, weakened bones, and other related conditions. Scurvy is a common disease that you know. Ang scurvy ay deficiency in vitamin C. Is vitamin C important for collagen synthesis as well? Yes. So deficiency of vitamin C could result to defective hydroxyproline formation and hydroxylysine formation, making collagen weak. So pag weak siya, very fragile yung collagen kaya mabilis masira. That's why in patients with scurvy, mabilis magdugo ang gums, for example. Okay? Kasi manipis or hindi stabilized yung structure ng collagen. So this is characterized by bleeding gums, as we said, swelling of joints, poor wound healing, and if not addressed immediately, that can cause death of a patient. So taken all together, that is the end of collagen. Remember, collagen is the first fibrous protein. Overall, naintindihan ba ang collagen at sakit na related sa collagen? What about the others? Kamusta kayo? Ha? Okay pa? Alright. Next, after we discuss collagen, proceed naman tayo sa pangalawa. Tatlo sila elastin tas keratin, right? So proceed naman tayo kay elastin. Elastin is an example also of a fibrous protein, and this is a connective tissue possessing rubber-like properties. So pag rubber-like, it can expand and it can go back to its original composition or original form. So yun yung characteristic ng elastin. Kaya nga siya elastin, elastic siya. So this is usually found in different areas of the body, particularly in the lungs, okay? In the lungs. In the lungs, and aside from that, in the walls of the blood vessels, that's why ang blood vessels at lungs, elastic sila. Okay? They can be expanded and they can get back to their original composition or original form rather. And then also in ligaments. So that's why I said that they can be stretched into several times and then they can recoil back to their original shape when relaxed. So yan ang property ng elastin. So regarding elastin, pag chinek natin ang elastin, this is composed primarily of small nonpolar amino acid residues. Again, this is composed primarily of small nonpolar amino acid residues. Wait lang for a while i-ano ko lang yung ventilation, wait lang. Ayan. Wait lang wait lang. Okay, still there? Tuloy natin. So I repeat, ang elastin, this is composed of small nonpolar amino acid residues. Like for instance, we have glycine. Glycine contains only hydrogen, right? And for alanine, methyl group, and valine naman, I think it has isopropyl functional group. So very small lang, but non-polar. So this is also rich in proline and lysine. However, in the case of elastin, little, okay, very little lang ang presence ng derived amino acids like hydroxyproline and hydroxylysine. Unlike ng collagen, marami. Okay? Unlike ng collagen, marami. So, another, we have interchain crosslinks form desmosine residue. So usually, kapag nagkaroon ng cross-linking ang iba't ibang amino acids, collectively, we can call them as desmosine. Now, what can you notice with the present amino acids in elastin? You can see that the present amino acid or amino acids are quite small in size. That's why flexible sila. Halimbawa, glycine, di ba? Important siya for flexibility. So for desmosine crosslink, again, this is present in elastin. This is an extensively interconnected rubbery network that can stretch and bend in any direction and will get back or recoil back to its original form, original appearance when relaxed. So that is desmosine. Remember, present usually ang elastin sa lungs, aside sa lungs, sa mga blood vessels and other connective tissues or ligaments. So in connection to that, i-discuss natin ang function ng elastin or yung connection ng elastin sa lungs and other compositions. So ito, medyo makaka-relate kayo. Check natin kung malalaman natin kung makaka-relate kayo dito. Okay? So first stop, as you can see on the figure, we have the lungs. And syempre tayo, habang nag-i-inhale tayo ng hangin, hindi lang naman oxygen ang nakakapasok sa lungs natin. Instead, there are other toxic substances that can enter through the lungs. And since these are toxic, the goal of the body is to remove those toxic compounds. So as we breathe, we not only take in oxygen, but other pollutants as well. So thankfully, on the animation that is shown, we have the color white ones. Yung color white, yung mukhang Pac-Man. Nakikita nyo white dyan? Nag-animate din ba sa Zoom? Nakikita nyo? Ha? So nakikita nyo yung puti. Ang tawag sa puting yan ay elastase. Okay? Ang tawag dyan ay elastase. So elastase is actually an enzyme that can degrade or that can remove those pollutants. Thankfully, we have elastase because elastase will get rid of, aalisin ng elastase yung mga pollutants na nakapasok sa lungs natin. However, we have to realize that we need to regulate elastase's action because normally, kapag ang elastase ay hindi mo napigilan, kapag hindi ito nag-stop, after killing, after removing the pollutants, it can destroy the lungs. So in short, it can destroy the elastin present in the lungs. Kaya nga siya elastase. It destroys. It metabolizes elastin. So ngayon, kung ang elastin sa umpisa ay beneficial at sa huli nakakasama, ang goal is i-regulate nga siya. So thankfully, meron tayong pang-regulate. Ang pang-regulate natin ay tinatawag na antitrypsin, okay? Or alpha-1 antitrypsin. So ang tawag doon ay alpha-1 antitrypsin. So this alpha-1 antitrypsin deactivates elastase after performing its job. So in Tagalog, i-deactivate ng antitrypsin yung elastase pagkatapos tanggalin ng elastase yung mga pollutants. Based on my discussion, essential ba ang antitrypsin sa katawan natin? Yes, okay, very good. Kaya lang ang problema, again, nakalagay dito by the way, without the antitrypsin, the elastase continues to consume anything, including healthy lungs. Healthy lungs because they contain elastin. Okay? So yun yung reason. Kaya dapat ma-maintain natin ang antitrypsin sa katawan. However, this is the problem. Ang problema usually is gumagawa tayo ng mga acts or activities that would lower antitrypsin level in the body. And the major reason kung bakit bumababa ang antitrypsin sa katawan ng tao kasi ang tao ay naninigarilyo, okay? Yan ang major reason kung bakit bumababa ang antitrypsin sa katawan ng tao. Question, kapag nanigarilyo at bumaba ang antitrypsin, do you think elastase could potentially destroy the lungs? Can it potentially destroy the lungs? Yes or no? Yes. Kasi ang elastase, active pa rin sila. Wala nang antitrypsin eh. Kaya nasisira ang lungs. Kaya kapag may chronic smoking, ang tendency, pwede magkasakit sa lungs ang tao at ang tawag doon ay emphysema. Okay, emphysema. Siguro familiar kayo sa respiratory disease na emphysema. So this is characterized by destruction of elastin in the lungs. Kaya nasisira ang mga alveolar walls made up of elastin. So therefore, kung sa emphysema, mababa ang antitrypsin, isang solution, isang treatment na ginagawa is mag-administer ng exogenous alpha-1 antitrypsin. Okay, mag-administer ng exogenous alpha-1 antitrypsin. Question, pag nag-administer ba ng alpha-1 antitrypsin, may available na in deactivating elastase? Ulitin natin. Kapag ba nag-administer ng alpha-1 antitrypsin sa tao, nagbigay, may available na ba sa katawan ng tao na mag-deactivate ng elastase? Yes or no? Yes. Okay, yes. Naintindihan niyo ba yung diniscuss ko sa connection ng elastase, ng antitrypsin at elastin? Naintindihan niyo yung tatlong yon? Okay, so very good. So, pag nag-administer ka ng alpha-1 antitrypsin, it will deactivate elastase. Therefore, mapoprotektahan ba ang elastin sa lungs? Mapoprotektahan ba ang elastin sa lungs? Yes or no? Yes. Okay, very good. So, mapoprotektahan. So, that's right. So, yan ang nangyayari kapag tayo ay naninigarilyo. Kaya, isang possible consequence ng chronic smoking is lung disease like emphysema. And alpha-1 antitrypsin administration is quite hard to procure or medyo expensive siya. Kaya the best way is to prevent. Okay? So yan yung ibig sabihin. So another figure that explains pathogenesis of emphysema. So for example, yan, tobacco smoking. So kapag naninigarilyo tayo, that can enhance neutrophil elastasis action that would eventually destroy the elastin present in the lungs. And because of that destruction, that could ultimately lead to emphysema. So ang problema lalo dito is, eto nga, nagkakaroon ng alpha-1 antitrypsin deficiency pag naninigarilyo. Kaya ang tendency, mas lumalala. So yan ang effect ng smoking. Gets? So done with elastin. So tapos na ang collagen, tapos na rin ang elastin. Third, we have keratin. So regarding keratin or alpha keratin, these are proteins that form, sorry, it must be form. These are proteins that form tough fibers. And regarding keratin, of course, this is usually found in hair, nails, and outer epidermal layers of mammals. And when we check the structure of keratin, keratin is actually rich in cysteine residues. Again, ang keratin is actually rich from Zyrel. May question si Zyrel, address muna natin. Sir, mas mabilis ba masira ang elastin pag secondhand smoking? Guys, tandaan natin, both the primary and first-hand and second-hand smoking, yaan ay magkukos ng destruction ng elastin. Bakit? Kasi same usok lang naman ang na-inhale and yung usok na yon may certain chemical compound, benzopyrene ang tawag, that would destroy the alpha-1 antitrypsin. So in short, ang sagot sa question mo, yes. Okay. Hindi naman mabilis, pero both. Okay? Both. Pero hindi mas mabilis. Both would be affected. Yon. Kasi yung mismong smoke ang responsible, yung nai-inhale na smoke, is responsible for the effect because it has a certain toxic compound. So moving on, so balik tayo, why hair and nails are insoluble. Pansin nyo ba, pag naligo kayo, hindi naman natunaw yung, yung buhok nyo. Pag naligo kayo, hindi naman natunaw yung kuko nyo, di ba? Kasi present siya or may present dyan na cysteine residues. Ang cysteine saan made up? Sa polar or non-polar residues. Anong meron dito? Polar or non-polar residues? What do you think? Polar or non-polar? Some answered polar. What about the others? O sige ha. Yung iba non-polar. O bakit polar yung iba? Or bakit non-polar yung iba? O isipin nyo ha. O sa mga nagsagot muna ng polar. O check natin kung rational yung answer. Pag polar ang sagot, ibig sabihin, natutunaw gamit ang tubig. Naligo ka. Ano pinaliligo mo? Tubig o langis? Siyempre tubig. Kung polar, gawa ang cysteine, ang buhok mo at kuko mo polar. So kung polar sila, pag naligo ka, wala ka ng buhok dapat, sumama dapat siya sa tubig. Pati yung kuko mo na wala, sumama sa tubig. Nagets nyo? So therefore, ano dapat na residues kung disulfide bonds ang present? Polar or non-polar? Non-polar. Very good, non-polar. Naintindihan? Naintindihan ba ako? Okay, very good. So present ang non-polar residues. And presence of cysteine residues or disulfide bonds in the hair would tell us that there are kinks present or may kinks, may kulot na makikita. So yung mga tao na nakulot ang buhok, mas marami sa kanilang disulfide bonds. So sa mga kulot na gusto magpa-rebond halimbawa, pwede nilang sabihin doon sa parlorista na pa-reduce po ng disulfide bonds. O yun yun. Pampastraight siya ng buhok. Okay? So yan ang keratin. So taken all together, that is the end. Finally, that is the end of fibrous proteins. O guys, recap tayo sa fibrous proteins. Pag pinag-usapan natin ng fibrous proteins, ano sila? Ano yung mga examples? Letter C, collagen. Pangalawa, elastin. And lastly, we have K. Ano yung K? keratin. Okay, keratin. That's right. Now, second group tayo ng physiological proteins. Sa second group ng physiological proteins, we have globular proteins. Remember, when we talk of globular proteins, they perform several roles. Number one, important sila for metabolism. Important sila for metabolism. And aside from that, nabanggit natin transport, transport, and then storage. Okay? These are important for metabolism, transport, and also storage. And also we have storage. So with this, sino si Rafael Tan? Tuwan-tuwa siya. So anyway, for metabolism, transport, and storage, ang focus natin for globular proteins ay yung functions na transport at storage. Transport at storage. And na-curious daw siya. Sa Zoom, pag nagsulat, makikita ng host kung sino nagsulat. Or kahit kayo ata makikita nyo. So anyway, okay lang, no problem. So anyway, sa transport and storage, this has something to do with oxygen. Okay, may kinalaman siya sa oxygen. Kaya iikot lahat ang discussion natin about oxygen. Unang tanong, importante ba ang oxygen for survival? Yes. So ngayon, pag-usapan natin, what's with oxygen? Of course, oxygen is very important for cellular respiration. This is very important for cellular respiration. So since ang pinanggagalingan ng oxygen ay lungs, kailangan siyang i-distribute into the different parts of the body because all cells need oxygen. However, if we think of, makinig ha, may kwento to, may kwento, para mag-gets nyo yung gap, yung problema. So, ang problema sa oxygen is, oo, kailangan siya ng katawan, pero mahirap siyang ipamigay. Ang hirap niyang ipamigay. Ano ang mga dahilan? Unang problema sa oxygen is, hindi siya humahalo sa aqueous solution. Ano bang example natin ng aqueous solution? Plasma, dugo. Okay, yung dugo. Ang dugo natin ay aqueous, ang oxygen hindi siya humahalo doon. So kung hindi siya humahalo, hindi siya basta-basta madidistribute kasi hindi sila nagkakasundo ng dugo eh. Okay? Pangalawa, kung sakali mang sumama siya sa dugo, hindi significant yung quantity, hindi ganun karami ang nakakarating sa tissues. So kung hindi marami ang nakakarating sa tissues, mamamatay din yung mga cells, right? And then pangatlo, distance dependent ang oxygen pag tinatransport. So lumayo lang ng konti sa cells, hindi na makakarating. Okay? So yun yung problem. Ulitin natin, importante ang oxygen, yun nga lang, kaya nga lang, mahirap siyang ipamigay. Okay? Yun ang problema. Next, ano ang pwedeng maging solusyon? Ano ang pwedeng maging solusyon? Ang pwede nating solusyon dito is kung ang oxygen, nasusundan pa ba ako? Nasusundan? Okay. Kung ang oxygen ay mahirap ipamigay ng mag-isa, ang goal is dapat dagdagan siya ng proteins. Okay? Dagdagan siya ng proteins. And pag sinabi nating proteins, ito ay binubuo ng mga amino acids. Right? Binubuo ito ng mga amino acids. Kaya lang, may problema na naman. Okay? Merong na namang problema. Ang problema is, yung lahat ng amino acids na makikita natin sa isang protein, lahat ng 20 standard amino acids, wala sa kanila ang may kakayahang mag-bind sa oxygen reversibly. So ang point doon is, oo, makukuha nila yung oxygen, pero sa protein na forever yon. So problema na naman siya. Di ba dapat pagkuha ng protein doon sa oxygen, pagdating sa tissue, ibibigay, i-give off. Pero ang problema sa mga amino acids, wala sa kanila ang nagbibigay after. Kanila lang yon forever. So problema na naman siya. So therefore, ano ang susunod na goal? Ito yung goal number 1, okay, dun sa oxygen. Ngayon, anong susunod na goal? Nagigets ba yung kwento? Ha? Para magets nyo kung bakit ganun sa dulo, okay? Bakit may heme protein. So, pangalawang goal is, kung ang protein itself ay hindi nag-give off easily ng oxygen, kailangan ang protein kabitan mo ng tinatawag na prosthetic group. Okay? Kailangan kabitan mo siya ng tinatawag na prosthetic group. So pag sinabi nating prosthetic group, again, pag sinabi nating prosthetic group, ito ay non-protein portion, a non-protein portion na may certain physiological role in the body. So in this matter, ang mga prosthetic groups usually mga metals, okay? Mga metals. Again, mga non-proteins. These are transition metals or mga metals. So sila ang mga prosthetic groups. Ang mga prosthetic groups, very essential sila because they have strong tendency to bind oxygen. Ang example natin ng prosthetic groups ay iron, ferrous ions. Again, ferrous ions. I repeat, ang common ay ferrous, as in 2 plus ha, 2 plus ferrous ions. Yan yung very common. Kaya pansin nyo, yung remedy sa baba, nakikinig pa ba? Nakikinig? So ang remedy sa baba, incorporation of transition metals that have strong tendency to bind oxygen and then eventually give off, ibibigay. So question, pag nagdagdag ng prosthetic group, ito ba ay masosolusyonan ang problema with proteins alone? Masosolusyonan ba nila yung problema pag protein lang ang nagdala ng oxygen? Yes or no? O, ulitin natin ha. Ulitin natin. Ulitin natin yung flow. Ang oxygen ba mag-isa? Makinig ha. Nakikinig kayo? Nakikinig? Ulitin natin. Makinig mabuti. Huy, nakikinig? Ulit. Ang oxygen, mahalaga sa katawan ng tao. Kaya lang hindi siya kayang ipamigay basta-basta. Kasi ito ay hindi humahalo sa dugo easily. So, ang solusyon dyan, dapat may mag-transport sa kanya ng mga proteins. Ang proteins ay gawa sa amino acid. Kaya lang ang problema sa amino acid, kahit isang amino acid, wala sa kanila ang may kakayahan na dumikit sa oxygen reversibly. Kaya ang solusyon, instead na protein ang dumikit, kailangan may kasama ang protein na didikit sa oxygen at siya ang magbibigay eventually. Anong tawag sa kasama ng protein na hindi naman protein pero doon didikit ang oxygen? Anong tawag doon? Prosthetic group. Prosthetic group. Ano ang mga members ng prosthetic group? Mga transition metals. Ang pinaka-common, anong ion ang pinaka-common? Ferric o ferrous? Ferrous. O ulitin ko, naintindihan nyo na ba? Okay. Now, mag-focus tayo sa prosthetic group. Mag-focus tayo sa prosthetic group. Remember, ang prosthetic group, importante siya para dumikit ang oxygen. Kaya lang ang problema, may problema na naman sa prosthetic group, okay? Ang problema sa prosthetic group is siya ay may capability siya na ma-oxidize. So kapag na-oxidize ang transition metal, ang tendency, so oxidation of transition metal, that can result to stress, oxidative stress. So that can result to oxidative stress o magiging reactive siya. So anong magiging implication kapag nag-result sa oxidative stress pag na-oxidize ang prosthetic group? Masisira ang cells. Nakakapatay ng cells yan. So problema na naman yan. Okay, problema. So to be able to address that problem, kailangan ang gawin natin is to shield. Okay, to shield or to cover the prosthetic group. So ang next goal natin is to shield or to cover the prosthetic group. So yan yung ating objective o yung ating goal. Okay? To shield or to cover the prosthetic group. So by covering the prosthetic group such as iron, hindi siya magiging reactive. Okay? Hindi siya magiging reactive. So ano ang ibig sabihin yan? So ito yon. How can iron be less reactive? I-shield natin. So for example, this is iron. So ibabalot natin siya ibabalot natin siya sa isang specialized organic ring structure. Ang tawag natin sa organic ring structure that covers iron ay protoporphyrin. Protoporphyrin. So ang tawag doon ay protoporphyrin 9. So ang tawag ay protoporphyrin 9. So para hindi less reactive yung iron, kailangan itago siya using the organic ring called protoporphyrin 9. Now, makinig, ang tawag natin pag magkasama yung iron at yung protoporphyrin 9, so prosthetic group plus the protoporphyrin 9, ang tawag dyan heme. Okay? Ang tawag dyan ay heme. So, pag tinago natin ang iron using the protoporphyrin 9, hindi na reactive yung iron. At pag may iron, makakadikit na yung oxygen. Tama? And then the proteins themselves, sila ang magta-transport. Kaya may tinatawag tayong heme. Okay? May tinatawag tayong heme. So heme is a protein-bound prosthetic group wherein at the center, we can see iron in its anong state? Ferrous state. Okay? Nasa ferrous state. And take note at the center, we have iron in ferrous state having 6 coordinated bonds. So pag sinabi nating 6 coordinated bonds, yung iron sa gitna, meron siyang 6 na kapitbahay. Okay, meron siyang 6 na kapitbahay. So una, yung 4, nandiyan pa kayo? Tapusin ko lang yung itsura ng heme tapos continue tayo bukas. Nandiyan pa kayo? Okay. So ilan ang coordinated bonds ng iron? How many? 6. So 4 doon is for nitrogen. Okay, nitrogen. So anong goal ng 4 na nitrogen? 4 na nitrogen na nakadikit sa iron. So that would make iron less reactive. Okay? So that would make the iron less reactive. Yun nga ang goal, di ba? Ikulong yung iron. So para hindi siya reactive, kailangan ng nitrogen 4. Now, what about the 2 remaining coordinated bonds? This will be answered later, okay, sa mga succeeding slides. Masasagot natin ang 2 coordinated bonds. Meron siyang reason. And then finally, ito muna yung final slide for today. Finally, those proteins containing heme are called as globular proteins or globular heme proteins. So meron tayong 2 klaseng globular proteins. So number 1, we have myoglobin. And then second, we have hemoglobin. Again, number 1, we have myoglobin. And then second, we have hemoglobin. So yan ang 2 types. Tandaan ang myoglobin, meron siyang isang heme. And then ang hemoglobin, meron siyang 4 na heme. Again, for myoglobin, there is 1 heme. And then hemoglobin, may 4 na heme. So ibig sabihin, halimbawa, this is the structure of myoglobin. Isang heme ang makikita natin. Question, may tanong ako, kada heme, ilang iron ang makikita? Ilang iron ang makikita? 1, 2, o 3? 1 lang. Very good. Ilan ang kapitbahay ng iron? 6. Di ba? 6 ang kapitbahay niya. 4 doon. Alin ang kapitbahay niya? 4. Nitrogen. Question, ano yung natitirang 2? Yung natitirang 2, ito yon. So first, we have histidine. Yung isang nakadikit ay isang histidine residue. Kaya ang nakakabit doon ay isang histidine residue. And ang tawag doon ay proximal histidine. Okay? So ang tawag doon ay proximal histidine. Next meeting, magigets nyo bakit proximal ang tawag. Sa ngayon, kabisaduhin nyo muna, yung isang coordinated bond ay proximal histidine residue. And then, yung natitira, yung huling coordinated bond para kay oxygen. Anong sabi ko? Tama ba na sa prosthetic group talaga dumidikit ang oxygen kanina? Ha? Sa prosthetic group pa dumidikit yung oxygen as we discussed earlier? Yes or no? Yes. Kaya for the nth time, pag binalikan natin ang structure ng, paulit-ulit to para matandaan nyo, ang structure ng heme, yung nasa gitna, iron, and then apat nitrogen, isang histidine, at isang oxygen. Isang oxygen. Yan ang kapitbahay ng iron. Okay? Last question for this meeting. If these are the coordinated bonds, i-relate natin siya sa diniscuss ko kanina. Kung ang myoglobin ay may isang heme, ilang iron meron siya? Isa. So therefore, ilang oxygen ang kayang dumikit sa kada piraso ng myoglobin? Isa lang din ba? Yes or no? Okay, yes. So isang oxygen lang. And then, ang hemoglobin, ilang heme meron siya? Apat. So kada pirasong hemoglobin, ilan ang didikit na oxygen sa kanya? Apat. Okay, apat. Kaya bawat hemoglobin, apat na oxygen molecules ang didikit kasi ang ratio dyan, 1 heme is to 1 oxygen. Okay? 1 heme is to 1. Naintindihan ang ratio? Okay. So siguro kota na tayo for today. Hanggang dito muna tayo ngayong araw. So recap tayo saglit bago ko kayo i-dismiss. Sa myoglobin, isang heme. So isang oxygen and sa ano yun, myoglobin. Sa hemoglobin, apat. So far ba na-gets ang ibig sabihin ng heme? Ang heme ba ay may iron? Ha? Ang heme ba ay may iron? Yes. Meron ba siyang protoporphyrin? Alam niyo yung protoporphyrin, di ba yung malaking ring? So, anong importance ng protoporphyrin? Tama ba na siya ang responsable para mag-bind ang oxygen? Yes or no? Ulit, makinig. Ang protoporphyrin ba ay importante para mag-bind ang oxygen? Yes or no? Bakit no? Ano ba talaga, Rafael Tan? Parang ngayon ko lang narinig yung pangalan niya. Parang mga nakaraang meeting, hindi ko siya napapansin. Anyway, bakit? Yes or no? Ulitin ko yung question na. Di ko kayo di-dismiss pag di kayo sumagot. So ulit, kapag protoporphyrin 9, anong function? Tama ba na ang function niya ay makabind ang oxygen? Yes or no? Why? Anong purpose niya? To make iron what? Less reactive. Very good. Yung sinabi ko kanina, kaninong function yon? Kay iron. Kumbaga, ang point dyan is, ganito yung hierarchy ha, para lang wag nyo makalimutan. Ang hierarchy nung sinabi ko kanina is oxygen. Tignan nyo na sa PowerPoint, oxygen. Hindi siya humahalo. Kaya kailangan may protein na magdadala. Ang problema sa protein, hindi siya nagbibigay ng oxygen ultimately. Susuluhin niya. Kaya kailangan may dumikit sa protein na iron, na prosthetic group. Kasi dyan didikit ang oxygen. Kaya lang, ang problema, si iron, pag expose siya, pwede siya mag-cause ng oxidative stress, killing cells. Kaya dapat may magtago sa kanya na malaking ring para pag nag-react ang iron sa oxygen, tago ang iron, and then may protein na magta-transport lahat ng ito. Ang tawag pag lahat-lahat to, heme protein ang tawag dyan, heme protein. Kapag walang protein at walang oxygen, pag iron at protoporphyrin lang, heme lang ang tawag. Heme lang ang tawag. Nagigets nyo ako? Ha? Kapag iron lang, anong tawag pag iron lang? Prosthetic group. Okay? Naintindihan? So hanggang dyan muna tayo for today. Meron bang naintindihan ngayong